Hi, my name is JC Farner, and my project was on comparing changes in secondary plant metabolites in response to drought stress. So the overall takeaway from this meta-analysis was that phenolics content increased while flavonoid content decreased in response to drought stress. And you can see that here in the graph. There's an average increase of around 240% in phenolics content and a decrease of around 7% in flavonoid content in response to drought. And then a t-test was used to evaluate the difference between these two average values and yielded a p-value on the order of 10 to the negative 48, which does indicate statistical significance here. Now, one possible interpretation of this data is that perhaps phenolics are more universally important in drought stress or a drought response in plants. Across the three different data sets used, there are around four general plant types: grasses, trees, shrubs, and the leaves of tuberous root plants. So perhaps flavonoids just aren't as universally or generally important across these different plant types where phenolics are. And then just as a little bit of background here, it's important to note that flavonoids are actually a subset of phenolics. Both are secondary plant metabolites, but you see here in this image, you have a phenol in the middle here, you have the different phenolics that it can become, and then flavonoids are a subset of those phenolics. Now, what's important about these two metabolites is they're both fairly broad categories involved in plant defenses, and that can be against herbivores, pathogen, drought, and other stresses. But to produce these secondary plant metabolites, it's pretty energetically costly. So generally, what will happen in plants is specific stresses, such as drought, will trigger the production of these metabolites, and this is what's known as an inducible response. Now, what's key about this induced response is that there are a finite amount of resources and energy available to the plant, such that if it has to upregulate its defense mechanisms, that comes at the cost of growth and reproduction. And you can see that better here in this image. You have abiotic stress, such as drought, triggering a stress response, leading to an increase in what are called reactive oxygen species, which are related to secondary plant metabolites, so you could just pretend that that's what that says there. And that's accompanied by a reduction in plant growth, yield, and photosynthetic activity. Now, this becomes a direct negative consequence on the organisms that subsist off of these plants, whether that be in a natural ecosystem or in agriculture. So reduced crop yield, reduced crop quality, or just reduction in biomass in the primary producer level of an ecosystem is going to have some pretty negative impacts on both agriculture and ecology. So further research evaluating how and why this occurs will hopefully lead to more sustainable solutions for the future of conservation work and crop management as we continue to face these warmer and drier years. Now, how this data product was put together, there was 3 data sets with different plant types across each. Each data set offered metabolite content information for both drought and ambient conditions. And then this equation you see here was used to get a percent change, which controlled for any differences in units, whether that be like nanograms or milligrams. So the metabolite content for each phenolics and flavonoids was plugged into this equation for each plant that was available in the data set and turned into this percent difference. And that was fairly straightforward for data sets 1 and 2, but for 3, a program called WebPlot Digitizer was used to extract the average metabolite content for drought and ambient conditions off of a graph. Once this percent change data point was calculated for each trial in each data set, they were averaged together for each flavonoids and phenolics respectively, and that average yielded the graph you saw on the first slide, the 240% increase for phenolics and the 7% decrease in flavonoids. And then a statistical test was used to evaluate if there was a significant difference between these 2% changes. It was found that there was, as you saw with that fairly small p-value. And then these were the sources I used for both the images and the data sets. Thanks for watching.